Hi guys, this is Dr. Hamid, an ECV certified physician, and we are having a very high yield revision for the whole microbiology chapter for the USMLE Step 1 and Step 3 exam. So let's dive straight in. The cell envelope has like multiple layers, for example, the capsule, cell wall, and the plasma membrane, and it is different between the gram-positive and the gram-negative. Not all the layers are different, but there are some common layers and some different layers. First, the two common layers on the outer surface are the capsule and the slim S layer. So the capsule being on the outer surface has a very important role, which is the prevention of phagocytosis. So it covers the layers behind it and protects like the bacteria against the immune system by hiding what lies inside and behind it is a succeeding layers. So the capsule is essentially formed from a discrete polysaccharide layer. So some parts of the bacteria are formed from polysaccharides, others from protein, and others from peptidoglycan. You have to know that the capsule is made from polysaccharide and it serves as a major virulence factor. So the term virulence factor is very high yield because it is essentially the ways that the bacteria either attack the body or defend itself. So all of that is termed virulence factor, and the capsule hides the bacteria from the immune system and prevents phagocytosis, and this is considered the virulence factor. Next is the slim S layer. So you may think of it as a continuation of the capsule because it is also formed from polysaccharides, and like the sugar is sticky, for example the candy, and you can memorize it this way because it mediates adherence to surfaces, and this adherence is also a virulence factor because the bacteria, when it is adherent and sticky, can help it in, for example, colonization of indwelling catheters and the devices that inserted inside of the body. So the bacteria can stick to these catheters and cause infection, and this is related to the slim S layer. So here starts the first difference between gram-positive and the gram-negative bacteria, which is outer membrane protein. So the term outer membrane here is pretty unique to gram-negative, so there is no equivalent outer membrane for gram-positive. So the outer membrane is have also something unique, which is called lipopolysaccharide slash lipooligosaccharide. So this is very antigenic. So lipopolysaccharide is composed from lipid and saccharides from its name, and the lipid A component is the most antigenic. So the oligosaccharides and all polysaccharides are also antigenic, but the lipid part, which is called the lipid A component, is like the most antigenic of the gram-negative, and it induces tumor necrosis factor and interleukin 1, and hence causes like inflammatory reaction. So this is for the outer surface. So the through the wall, the outer membrane have something called porins. So these are essentially channels that transfer stuff across the outer membrane, and also it contains inner membrane. So the inner leaflet of the outer membrane consists of phospholipids. So this is related to the outer membrane. So again, this is purines and outer membrane proteins. So the outer membranes have like outer membrane proteins that are embedded within and have purines that transfer stuff across the outer membrane. So also most outer membrane proteins are antigenic, but it is highly tested in the exam to know that the lipid A component of lipopolysaccharide slash lipooligosaccharide is the most antigenic and induces tumor necrosis factor and interleukin 1 as a reaction. So this is outer membrane. If you like, this is gram-negative bacteria. This is the outer, like the whole cell envelope of gram-positives. So if you focus here, there is no equivalent for this outer membrane in gram-positives. So also we have a continuation which is called the periplasmic space. So The periplasmic space is a space between the cytoplasmic membrane and the outer membrane, and it's logically there is no periplasmic space in gram-positive because they don't have outer and inner membranes. They have one membrane, and from the definition of periplasmic space being between the cytoplasmic and the outer membrane, it is unique for gram-negative also. So it accumulates components like hydrolytic enzymes such as the beta-lactamases. So the beta-lactam antibiotics tries to fight the bacteria, and they are not very efficient against gram-negatives because the periplasmic space, which is unique to gram-negative, contain like some hydrolytic enzymes such as the beta-lactamase. So this is regarding the gram-positive and the gram-negative bacteria. First, the difference, which is the outer membrane. Next, the cell wall. So this again here, this is the outer membrane for gram-negative, and it has purines and other transmembrane proteins, and it contains the inner layer of it contains phospholipids versus lipopolysaccharides / lipooligosaccharides. And this is related to the outer membrane. Next, we have peptidoglycan cell wall. So the cell wall, you can think of it in terms of like the skeleton. So of course, both gram-positive and the gram-negative will have a skeleton, and this is what gives them shape and like makes them like resist the harsh environment. So it is essential for both of them, but there is a kind of difference because the gram-positive also it does not contain an outer membrane. It is compensated by having like on the other hand, it's not like kind of compensation, but on the other hand, it has like a thick peptidoglycan wall, like versus the thin peptidoglycan wall in gram-negative. So the gram-negative have a thin peptidoglycan wall versus the thick peptidoglycan wall in gram-positive. So it is a sugar backbone with peptide-sided chain and cross-linked by transpeptidases. So you have to know that it is peptidoglycan, so it has like peptides and the glycan. So the sugar is the backbone, and the peptides are inside chains, and the crossage and the linking is through the transpeptidase, and it is a target. You have to know that, and it is a target of antibiotics, so you have to know the transpeptidase very well, and this provides rigid support and osmotic protection, so as we've discussed. Next is the cytoplasmic membrane. So the cytoplasmic membrane is also common between gram-positive and the gram-negative. So the peptidoglycan. So before we go next to the cytoplasmic membrane, I want to say is that the peptidoglycan cell wall lies in the periplasmic space in gram-negative. So we've discussed that the periplasmic space is unique to gram-negative, and there's like thin peptidoglycan layer lies within this periplasmic space versus the thick peptidoglycan layer in the gram-positive. And the cytoplasmic membrane is a phospholipid bilayer that we've studied in the biology, which is containing the penicillin binding proteins, oxidative enzymes, and the site of the cell wall synthesis. So this is the true membrane of the both gram-positive and gram-negative, and it is the site of a cell wall synthesis. So the cell wall lies on the outside of it, and it is like kind of a precursor or the site for its synthesis. So you have to know that, and it contains the penicillin binding proteins, which is very high yield. So there is remember we've discussed the lipopolysaccharide versus the lipooligosaccharides. So the lipopolysaccharides and versus they have like kind of a rule in the gram-negative by inducing inflammation. So inducing inflammation in gram-negatives is mainly dependent on this and the lipid A component. We've got an equivalent here, which is the lipotecoic acid. So this is the lipotecoic acid. It extends from the inner membrane all the way out. So here this is lipotecoic acid. It extends from the inner membrane all the way out, and it is unique for gram-positive and induces also tumor necrosis factor alpha and interleukin-1. So you can think of it as equivalent for lipopolysaccharide slash lipooligosaccharide in gram-negatives. And so we've discussed like the outer structure of or the envelope of both a gram-negative and a gram-positive. So we are going to discuss the gram stain. So it is a kind of a quick way to identify the bacteria before we do culture. And there are like multiple stains. So the most famous one, of course, is a gram stain. So you have to know the idea behind it. So it depends mainly on the peptidoglycan thickness. So we start with a crystal violet, and we pour it on this culture, and the peptidoglycan layer absorbs this crystal violet. So remember from the past slides that like gram-positive have a thick peptidoglycan layer, and hence it is gonna absorb this crystal violet, versus the like gram-negative, which have thin peptidoglycan layer, is not gonna absorb it very well, and it is gonna appear pink with safranine, like the counter stain. You don't have to memorize these stain names, but this is the idea behind it. So the thick layers retain the crystal violet iodine, so the thin layers lose it, and by the ethanol, when we wash this stuff with ethanol alcohol, and then when we do the counter stain, and it gives like these gram negatives their pink color. So the gram positive results the retain primary stain and appear purple blue under the microscope, versus the gram negative, they lose the primary stain and they take the counter stain and appear pink. So this is the first line lab test for bacterial identification and guiding empirical antibiotic therapy. So it's kind of quick because the culture takes time, so we may do first the gram stain. So we have a problem here because this test depends on the thickness of the peptidoglycan layer. So some bacteria have like a problem here because the Treponema and the Leptospira, for example, they are too thin to be visualized at the beginning, the Treponema and Leptospira. And others, they don't have a cell wall because the peptidoglycan lies in the cell wall of the bacteria, such as the Mycoplasma and Ureaplasma. So it is kind of irrelevant here to do gram staining for Mycoplasma and Ureaplasma, and others having like a high lipid content, such as the Mycobacteria. So you don't like here of Mycobacterium tuberculosis, for example, having like gram stain because they have high lipid content and they resist the dye. So others are primary intracellular. So being primary intracellular, we will have like hard time identifying them with a gram stain, such as Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia. So there are like some organisms that are like obligatory to be intracellular, and others like can like facultatively be intracellular or extracellular. So this is very important that we are going to talk about this later. And next, there are like specialized stains. So these specialized stains like solve the problems for the gram like stain. So we talked about how limited a gram stain for identifying organisms. Not all the organisms can be identified by gram stain. So we are going to discuss some of the other stains, for example, the Giemsa stain, used to visualize other. So remember, the chlamydia was intracellular and rickettsia. So these organisms, some of them have like problems being identified by gram stain, so we do other stains. So also a very high yield to know that the Plasmodium which is a causative organism for malaria, can be identified by this stain, which is called the Gibbs stain. So it is essential for identified malaria parasites. So these are parasites like trypanosoma here. So these are trypanosoma here, which are identified by Gibbs stain. So you have to know the stain and some of the most famous organisms, at least not all of the organisms. So here is a Gibbs stain. You have to memorize the Plasmodium and trypanosoma, and for example like chlamydia. And there is other stain which is called BAS, which is the periodic acid Chef stain. So the target for this stain is the glycogen. So this stains is a glycogen and the mucopolysaccharides, and it is the primary diagnostic tool for some disease called Weibel's disease, which is a disease of the GIT called Bacterial Weibelii. We are going to discuss these diseases later on in the as we are explaining stuff. So other stains which are going to discuss is the Zehel-Neelson stain. So the Zehel-Neelson stain, remember that the mycolic acid was like we kind of like resistant for the gram stain, so we solved that by this Zehel-Neelson stain. So this Zehel-Neelson stains the mycolic acids in the cell wall used for the mycobacteria and the nocardia. Very high yield to memorize this stuff. The mycobacteria is identified by the Zehel-Neelson stain, not the gram stain, and also the nocardia. These organisms appear like bright red against a blue background. And other stain which is also highly tested is the India ink stain. So you have to memorize this is the stain for Cryptococcus neoformans. So it is kind of a counter stain because you are staining the whole thing, and the capsule is thick in the this thick polysaccharide capsule in the Cryptococcus neoformans resists the stains, so leaves the clear halo against a dark background. So this clear halo against a dark background in Cryptococcus neoformans is because this capsule is thick and resists India ink. So other stain, which is the silver stain, is very high yield for Helicobacter pylori. So this is a Helicobacter pylori here, these organisms here in the GI tract, and it is stained by the silver stain. And also the fungi, we use the silver stain for the fungi coccidioides and the Pneumocystis reversi and Aspergillus fumigatus. So Pneumocystis reversi, Aspergillus fumigatus, and coccidiosis identification, and also Helicobacter pylori. So these are some of the uses for the silver stain. Next, so we've talked like the stains, which is a kind of a quick way for identification for these bacteria. Then we may do culture. So the culture we need to do culture to identify the specific organism to fight it and target it with the specific antibiotics. So this takes some time, and so it is not kind of alternative for the gram stain, but they are complementary to each other. So there are some organisms that are fastidious, so they need some support. For example, the Haemophilus influenza. So the Haemophilus influenza requires a chocolate agar, which is like after we lyse the red blood cells, it is called the chocolate agars, and the slices of blood cells provide us with some supportive nutrients such as factor 5, which is NAD+, and factor 10, which is a hematin. So these are released from the lysed RBCs, and this supports the growth of H. influenza. So this is a chocolate agar. You have to memorize the chocolate agar and the organism that grows in the chocolate agar, which is the Haemophilus influenza. Other important and very high yield organism is the Neisseria. You know, I have Neisseria gonorrhea and Neisseria meningitis. So these are the two different high yield Neisseria species. So they selectively are grown on Cyre-Martin agar. So the idea behind the Cyre-Martin agar is we create like a selective environment for this Neisseria by fighting other organisms. So we start by eliminating the fungi with nystatin. So this is first, and second we Neisseria are gram-negatives, so we eliminate the gram-positives by vancomycin. So we this is the second antibiotic that we are adding to the Cyr-Martin agar. And third, we eliminate other gram-negatives, so other than the Neisseria with trimethoprim, colistin. So we had vancomycin to eliminate gram-positives, fungi with nystatin, and the trimethoprim colistin to eliminate other gram-negatives other than the Neisseria. And other media that is very important is the boarded ginkgo agar. So it's kind of like a potato extract. So this potato extract is like media with nutrients to help grow Bordetella pertussis, which is a cause for a whooping cough that we are going to discuss later. Or can we use like Regen-Lewy media also to grow Bordetella pertussis, which contains like charcoal, blood, and antibiotics. So this is for Bordetella pertussis causing whooping cough. And also the Corynebacterium diphtheria have like a special culture where like telluride agar or leafriar media, and this is black colonies of diphtheria on telluride agar. So this again, so you have to memorize that telluride agar with the Corynebacterium diphtheria. And lastly, the Mycobacterium tuberculosis, after we like identified it with special stains, and we need to culture it. So it is cultured on the LJ media or the Middlebrook medium, and it requires long incubation period. So we start like treating and don't wait essentially for the cultures, as we are going also to discuss later. So we are going to talk about something which is called the lactose fermenters and E. coli. So this refers to the Enterobacteriaceae family. So these are gram-negatives and also Pseudomonas. So the whole idea behind it is after lactose fermentation, the media is turned into pink, versus the other organisms that are non-lactose fermenting, which are colorless here. So these organisms turn to pink because they are lactose like to differentiate like them based on like fermentation, and this fermentation is translated into a certain color, which is a pink in case of lactose fermentation. And azine blue agar is used for identification for E. coli, as the E. coli appear with a green metallic sheen here. So this green metallic sheen here is the E. coli after being grown on the azine methylene blue. So lastly, we are like talking about some organisms such as Legionella. Important organisms can be grown on a charcoal yeast extract agar with cysteine and iron. So these are some of the nutrients: cysteine, iron in charcoal yeast extract agar to support the growth of Legionella, and the sabarod agar. So the sabarod agar contains high acids to kill the bacteria and favor like the growth of fungi. And lastly, the Mycoplasma pneumoniae requires like eton agar, and it needs like cholesterol, so it needs cholesterol for membrane stability. So these are some of the special media that support certain organisms to grow. And I hope you enjoyed this, and see you later in next episodes in this series, as we are completing this very high yield revision for the microbiology chapter for the USMLE exam. Thank you guys.